Welcome back to the channel. In this video, I want to show you how to design primers for PCR. Please check out my personal blog where you can find a detailed written version of the video. Thank you for checking it out. The link is in the video description. I want to start this video with an example. Imagine that you work in a research laboratory and you want to investigate a target gene that is 300 base pairs long. And what you want to do is you want to reduce the size of this target gene to 200 base pairs. And you decide to do a PCR in order to achieve this. So you're interested if the gene of interest behaves differently if you cut off 100 base pairs. And you want to investigate then later the protein. And what you also want to do is you want to reinsert this resulting 200 base pair long gene back into your original plasmid. And you choose a cloning method where you use restriction enzyme sites in order to achieve this. So if we write out our target gene as a string of DNA base pairs, it would look like this. And if we do a PCR, it should look like this in the end. So you want to have BOMH1 at the 5' end and ECORI1 at the 3' end. And you also want to introduce a stop codon. Now the question is, how do I design the primers? Before we start with the primer design, I want to give you some rules that should always be applied when designing primers. The first rule is that it should be between 18 to 24 bases long. The second rule is that the melting temperature should be between 50 and 60 degrees Celsius. And primers should have a similar annealing temperature. What's also very important is that primer pairs should not have complementary regions, meaning that they should not bind to each other. So if we now start designing the primers and we start with the forward primer, it is very easy. The forward primers is basically just copy paste the first line of your gene of interest. So in this example, you take the first 18 to 24 base pairs starting at the 5' end and copy and paste this. That's it. That's your forward primer. And it would look like this. Then for the reverse primer, you have to think in an anti-parallel complementary way. And what does this mean? This means that we start at the 3' end and we again take 18 to 24 bases and we take the complementary base pairs. So this means that instead of using a C, we use now a G. Instead of using the T, we now use an A, and so forth until we end up with a primer that should look like this. So to sum up, the forward primer is basically just copy paste the first line of your gene of interest, and for the reverse primer you take the first 18 to 24 bases starting at the 3' end and writing the complementary base pairs, and that's it. And then once you have your primers, you can now take into consideration the annealing temperature. The annealing temperature has to be similar for both primers. And you can achieve this by either adding base pairs or subtracting base pairs. And you can use software such as Serial Cloner, which is free, to calculate a similar TM for both of them. And once you have a similar TM for both of them, you can then decide what kind of polymerase you're going to use. So for example, a DreamTag or a Q5 or an S7 polymerase. And once you know which polymerase to use, you can go to the respective company site and then copy paste your primer pairs and also the concentration you want to use. And they will automatically calculate an annealing temperature that works for both primers. And actually, I think that everybody does it like this. You can also do it by hand, but it's kind of complicated and it takes a lot of time. So I would just recommend to follow these steps. So basically design your PCR protocol, find out what kind of concentration you're going to use, find out the GC content of your primer, and then adjust the annealing temperature using some kind of software. So where you add or subtract base pairs, and then once you have a similar TM for both of them, you basically just go to the company side and copy paste your forward and reverse primer, add the concentration, and it will give you your annealing temperature, which is perfectly adjusted for the polymerase you're going to use. And that's it. Then one last thing that's very important is that you should exclude the restriction enzyme sites and also the stop codon from this calculation. So you just copy paste the actual primer. So in this example, this would be the red part of the primers. And then last but not least, what you should also do is that you should add sitting regions. So sitting regions are specific for restriction enzyme sites, and you can Google them online. You Google sitting region for BAM-H1 or Ricori1, and it will give you the corresponding sitting region. And sitting regions are important because they allow for better cutting. So if we add the sitting regions, we make sure that the restriction enzyme sites are actually part of our target gene. And that's it, guys. Thank you so much for watching. I hope that you enjoyed the video, that you learned something. Please like and subscribe, and I see you guys in the next video. Bye.